In Chapter 14, we are going to cover DNA structure and function. Our current understanding of DNA began with the discovery of nucleic acids, followed by the development of the double helix model. In the 1860s, a physician named Frederick isolated phosphate-rich chemicals from white blood cells, commonly known as leukocytes. And he named these chemicals, which would eventually be known as DNA, nuclein, because they were isolated from the nuclei of cells. Later on in 1928, a British scientist named Frederick Griffith reported the first demonstration of bacterial transformation. And this is a process in which external DNA is taken up by a cell, and that causes the cell to change its morphology as well as its physiology. Griffith conducted his experiments with the bacteria Streptococcus pneumoniae. And this is one of the bacteria that's commonly known to cause pneumonia. Griffith worked with two strains of this bacterium called the rough strain, that's denoted by the capital letter R, and the smooth strain, that's denoted by the capital letter S. The R strain is the non-pathogenic strain, so this is the one that does not cause disease. The S strain is the pathogenic one, so that is the one that is responsible for causing the disease. And the S strain was unique because it actually had a capsule outside of its cell wall. And the capsule allows the cell to escape the immune response of the host mouse. And what he did was he injected the S cells and the R cells into mice to see whether they lived or whether they died. So when Griffith injected the living S strain into mice, they died from pneumonia. But when he injected the live R strain into mice, they survived. In another experiment, when he injected mice with the heat-killed S strain, they also survived. This experiment showed that the capsule alone was not the cause of death. In a third set of experiments, what he did was he mixed some of the live R strain and the heat-killed S strain and injected that cocktail into the mice. And to his surprise, the mice actually died. So upon isolating the live bacteria from the dead mice, only the S strain of the bacteria was recovered. When this isolated S strain was injected into fresh mice, the mice died. So what does that mean? Well, Griffith essentially concluded that something had passed from the heat-killed S strain into the live R strain and transformed it into the pathogenic S strain. He called this the transforming principle. These experiments are now known as Griffith's transformation experiments. After Griffith's preliminary experiments, this kind of sparked an interest throughout the scientific field. So some other scientists wanted to investigate this phenomenon known as transformation. So Oswald Avery, Colin MacLeod, and Macklin McCarty did some experiments to further explore this principle. And what they actually did was they isolated the S strain from the dead mice and isolated the proteins and nucleic acids. So they took out the RNA and DNA as well as some proteins, as these were all possible candidates for the molecule of heredity. They used enzymes that specifically degraded each component and then used each mixture separately to transform the R strain. They found out that when DNA was degraded, the resulting mixture was no longer able to transform the bacteria, whereas all of the other combinations were able to transform the bacteria. This led them to the conclusion that the DNA was the transforming principle. And even though the scientific experiments of Avery, McCarthy, and MacLeod were relatively sound and demonstrated that the DNA was the informational component transferred during transformation, a lot of debate was still occurring and a lot of unknowns were still in the field. So Hershey and Trace wanted to further investigate this, and what they did is they used bacteriophages, which is a virus that infects bacteria. And we've talked about viruses briefly in previous chapters, but they have a relatively simple structure. They're not alive unless they go into a host, and they're merely made up of a protein coat called the capsid and a nucleic acid core that contains their genetic material. So what happens with the bacteriophage is it actually has the capability to infect the host bacterial cell by attaching to its surface, and then it injects its nucleic acids inside the cell. The phage DNA makes multiple copies of itself using the host machinery, and eventually the host cell bursts, which causes the release of a large number of bacteriophages. Hershey and Chase selected radioactive elements that would specifically distinguish the protein from the DNA in the infected cells. So they labeled one batch of phage with radioactive sulfur, which is denoted by the superscript 35S, to label the protein coat. Another batch of phage were labeled with radioactive phosphorus, which is denoted by superscript 32, capital P. Because phosphorus is found in DNA, but not in protein, the DNA and not the protein would be tagged in the radioactive phosphorus. Likewise, sulfur is absent from DNA, but present in several amino acids such as methionine and cysteine. And this is a schematic representation of Hershey and Chase's blender experiments. So what they did here is they used radioactive phosphorus in order to label DNA, and they used radioactive sulfur in order to label the protein coat. So then what they did was they infected bacteria with the phage. So this is the one infected with the radioactive phosphorus, and the one I'm going to circle now in blue is the one that's infected with the radioactive sulfur. So then the next step, what they did is they centrifuged. They spun the samples at very high speeds in order to separate the phage from the bacteria. So stage 3, this stage right here, is the centrifugation step. And then what they did is they cultured the bacterial pellet. So the pellet's going to form on the bottom of the test tube after centrifugation. And what they noticed was that bacteria infected with the phage containing the radioactive phosphorus labeled DNA produced the radioactive phosphorus labeled phage. But the bacteria that were infected with the radioactive sulfur labeled phage produced an unlabeled phage. So again, this is conclusive that only the radioactive phosphorus entered the bacterial cells, indicating that DNA is the true genetic material. And around the same time that Hershey and Chase were conducting their experiments, an Austrian biochemist named Erwin Chargaff examined the content of DNA in different species, and he actually found that the amounts of adenine, thymine, guanine, and cytosine were not found in equal quantities, and that the relative concentrations of these 4 nucleotide bases actually varied from species to species, but not within tissues of the same individual or between individuals of the same species. He also discovered something that he didn't really expect. He discovered that the amount of adenine equaled the amount of thymine, and the amount of cytosine equaled the amount of guanine. And that kind of goes with what we now know today, that adenine binds with thymine and guanine binds with cytosine, as far as the essential nucleotide base pairing goes. Furthermore, he found that different species had equal amounts of purines, which are adenine and guanine, as well as the pyrimidines, which are thymine and cytosine. But there were different ratios of A and T to G and C. And these observations became known as Chargaff's rule. Chargaff's findings proved immensely useful when Watson and Crick were getting ready to propose their DNA double helix model. And this slide here shows you the structure of the different nucleotides we talked about on the previous slide. So we see cytosine, thymine, adenine, and guanine. We didn't mention uracil yet because uracil is found in RNA, and uracil replaces adenine in RNA binding. So each nucleotide is made up of a sugar. So the sugar in DNA is the deoxyribose, and the sugar in RNA is the ribose. It's also made up of a nitrogenous base, so it's either going to be one of the pyrimidines or purines. And it's also made up of a phosphate group. In the 1950s, Francis Crick and James Watson worked together to determine the structure of DNA at the University of Cambridge. Other scientists like Pauling and Wilkins were also actively exploring this field. Pauling previously had discovered the secondary structure of proteins using X-ray crystallography. In Wilkins' research, there was another researcher named Rosalind Franklin who was using X-ray diffraction in order to understand the structure of DNA. Watson and Crick were able to piece together the puzzle of the DNA molecule on the basis of Franklin's essential data because Crick had also studied X-ray diffraction as well. And in 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Medicine. Unfortunately, Franklin had died, and the Nobel Prizes were not awarded to those who are dead at the time. And this picture here shows you the DNA structure that we still use and know today. So this is what Watson and Crick originally proposed, was that DNA is made up of two strands that are twisted around each other to form a right-handed helix. And the base pairing takes place between a purine and a pyrimidine on opposite strands. So remember in the previous slide I told you adenine is going to pair with thymine and guanine is going to pair with cytosine. And this is suggested by Chargaff's rules as well. So adenine and thymine are complementary base pairs, and cytosine and guanine are complementary base pairs. The bases are stabilized by hydrogen bonds. And this is really important for the test. Which bond pair has more hydrogen bonds? Adenine and thymine form 2 hydrogen bonds, while cytosine and guanine form 3 hydrogen bonds. So the bonds between cytosine and guanine are more difficult to break than the bonds between adenine and thymine, simply because the higher number. The two DNA strands are anti-parallel in nature, so what that means is the 3' end of one strand is going to face the 5' end of the other strand. The sugar and phosphate of the nucleotides form the backbone of the structure, whereas the nitrogenous bases are stacked inside like the rungs of a ladder. And each base pair is separated from the next base pair by a small distance, and each turn of the helix measures about 3.4 nanometers. So there are about 10 base pairs which are present per turn of the helix. Only the pairing between a purine and a pyrimidine and the anti-parallel orientation of two DNA strands can explain the uniform diameter. The twisting of the two strands around each other results in the formation of uniformly spaced major and minor grooves. This is a really cool and informative video on the Sanger method of DNA sequencing, so at this time, please pause the PowerPoint and check out this video. There are 3 suggested models of DNA replication, and they're depicted on the schematic here. So the first one is going to be the conservative replication. So what happens in conservative replication is the original parent double strands remain intact, and 2 completely new double helix strands are synthesized. So the 2 parent strands remain intact, and there are 2 brand new strands. This is the conservative DNA replication model. It's conserved because we conserve the initial integrity of the parent strands in the offspring. The next is going to be the semi-conservative method. And with the semi-conservative replication, the 2 strands actually come apart. So each acts as a template for new synthesis of a complementary strand. So you see one of the parent strands here, and you see the other one right here, but we see 2 new strands here. So this is semi-conservative because we are conserving some of the parent's genetic information in each of the replication mechanisms. And then we have dispersive. So in dispersive replication, both original parent strands are broken up into small pieces and incorporated into newly synthesized strands. So none of the strands are going to be identical to the parent's genetic information, because you can see here all of the new genetic information that is intertwined in the replication. So again, the parent strands are going to be broken up, and small pieces are going to be incorporated into these newly synthesized strands. So there is parental material there, but not an exact copy. 2 other scientists, Meselson and Stahl, were interested in understanding how DNA replicates. So what they did is they used E. coli, and they grew it for several generations in a medium containing a heavy isotope of nitrogen known as nitrogen-15, which gets incorporated into nitrogenous bases and eventually into DNA. So the E. coli was cultured, and then it was placed into the medium containing nitrogen-14, and then it was allowed to grow for several generations. After each of the first few generations, the cells were harvested and the DNA was isolated, and then it was centrifuged at high speeds in an ultracentrifuge. And during the centrifugation, the DNA was loaded into a gradient and then spun at high speeds of 50,000 to 60,000 revolutions per minute. Under these circumstances, the DNA will form a band according to its buoyant density. and this is the density within the gradient at which it floats. So DNA grown in the heavy nitrogen, or the nitrogen-15, will form a band at a higher density position. So it's going to form a band farther down the centrifuge tube compared to that one that's grown in nitrogen-14. So Meselson and Stahl noted that after one generation of growth in nitrogen-14, after they had been shifted from nitrogen-15, the single band observed was intermediate in position between DNA of cells grown exclusively in nitrogen-15 and nitrogen-14. This suggested either a semi-conservative or a dispersive model of replication. The DNA harvested from cells grown for 2 generations in the nitrogen-14 formed 2 bands. One DNA band was at the intermediate position between 14 and 15, and the other corresponded to the band of nitrogen-14 DNA. These results could only be explained if DNA replicates in a semi-conservative manner, and for this reason, therefore, the other 2 models were ruled out. DNA replication has been well studied in prokaryotes, primarily because of the small size of their genome, as well as the large variety of mutants that are available. E. coli has 4.6 million base pairs in a single circular chromosome, and all of it gets replicated in approximately 42 minutes, starting from a single site along the chromosome and proceeding around the circle in both directions. So this means that about 1,000 nucleotides are added per second. So the process is rapid and occurs without many mistakes. So how does DNA replication in prokaryotes get carried out? Well, it employs a large number of structural proteins and enzymes, and they each play their own critical role during the process. The first player we're going to talk about is DNA polymerase, also known as DNA pol. This adds nucleotides one by one to the growing DNA chain that is complementary to the template strand. The addition of nucleotides requires energy, and the energy is usually obtained from the nucleoside triphosphates, such as ATP and GTP. Like ATP, nucleoside triphosphates are high energy molecules that can serve both as the source of DNA nucleotides and the source of energy to drive polymerization. When the bond between phosphate is broken, the energy is released to form the phosphodiester bond between the incoming nucleotide and the growing chain. In prokaryotes, there are 3 types of polymerases. There's type 1, type 2, and type 3. It's now known that DNA polymerase 3 is the enzyme required for DNA synthesis. DNA polymerase 1 is an important accessory enzyme in DNA replication, along with DNA polymerase 2, which is primarily used for repair. So how does this whole machinery know where to begin? Well, it turns out that there are specific nucleotide sequences called origins of replication, or ORIs, where the replication actually begins. So in E. coli, which has a single origin of replication on its 1 chromosome, just like most prokaryotes do, this origin of replication is about 245 base pairs long and is rich in adenine and thymine sequences. So the origin of replication is recognized by certain proteins that bind to this site. An enzyme called helicase unwinds the DNA by breaking the hydrogen bonds between the nitrogenous base pairs. ATP hydrolysis is required for this process, and as the DNA opens up, Y-shaped structures called replication forks are formed. 2 replication forks are formed at the origin of replication, and these get extended bidirectionally as replication proceeds. Single-strand binded proteins coat the single strands of DNA near the replication fork to prevent the single-stranded DNA from winding back into a double helix. So this slide shows some of the first components of DNA replication. So as DNA replication begins, the DNA helicase, which is a large enzyme, is going to be responsible for separating the 2 strands of DNA so they can act as templates for replication. So the helicase is the unzipper. Single-strand binding proteins bind to each strand to stabilize and prevent them from reforming the double helix. Next, we're going to talk about primase. So primase is an RNA polymerase that binds to the single-stranded DNA and synthesizes a short RNA primer in the 5' to 3' orientation that is anti-parallel to the parental strain. This RNA primer allows for DNA polymerase to begin replicating the DNA. Topoisomerase, which we see over here, binds to the double helix upstream of the replication fork. So this is the replication fork right here, in order to prevent additional coiling by making small cuts in one of the DNA strands. The replication fork moves really fast. It actually moves at the rate of 1,000 nucleotides per second. And topoisomerase, as I mentioned on the previous slide, it prevents the overwinding of the DNA double helix ahead of the replication fork as the DNA is opening up. And again, it does so by creating these temporary nicks in the DNA helix and then resealing it. And because DNA polymerase can only extend in the 5' to 3' direction, and because the DNA double helix is anti-parallel, there's a slight problem right here at the replication fork. There, the two template DNA strands have opposing orientations. One strand's in the 5' to 3' direction, and the other strand is oriented in the 3' to 5' direction. So only one new DNA strand, the one that is complementary to the 3' to 5' parental DNA strand, can be synthesized continuously towards the replication fork. This continuously synthesized strand is known as the leading strand. The other strand, complementary to the 5' to 3' parental DNA, is extended away from the replication fork, and this is in small fragments known as Okazaki fragments. And these each require a primer to start the new synthesis. New primer segments are laid down in the direction of the replication fork, but each pointing away from it. The strand with the Okazaki fragments is known as the lagging strand. So how could E. coli replicate itself so fast and so efficiently without making mistakes? What is the proofreader? Well, that is DNA polymerase. So DNA polymerase is responsible for detecting and correcting errors during replication. So any mistakes that are made, DNA polymerase is going to detect them and repair them. In mismatch repair, the incorrectly added base is detected shortly after replication, and the mismatch repair proteins detect this base and remove it from the newly synthesized strand by nuclease action. Then the gap is going to be filled with the correctly paired base. So nucleotide excision is responsible for repairing thymine dimers. So sometimes when exposed to ultraviolet radiation or ultraviolet light, thymines lying adjacent to each other can form covalently linked thymine dimers. So in normal cells, these mutations are excised and replaced. And the schematic on the left-hand side shows you the process of the replacement. So again, anytime we have an ACE, that means to cut. So the nuclease is responsible for cutting the DNA on each side of the dimer and also helps to remove the single strand of DNA containing the dimer. The DNA polymerase we talked about on the last slide is responsible for detecting those mismatching sequences and helping with the repair. So the DNA polymerase is going to synthesize new DNA to repair the whole using the other strand as the template. And anytime you hear the word ligase, that means to join. So the DNA ligase is what is responsible for forming that final phosphodiester bond between the new DNA at the 5' end and the old DNA at the 3' end. Replication potentially shortens the ends of eukaryotic chromosomes. So the ends of linear chromosomes are actually maintained by the action of the telomerase enzyme. And on the next couple slides, we're going to talk more about the importance of telomerase. Telomerase is typically active in germ cells and adult stem cells. It is not active in adult somatic cells. So this is a picture of Elizabeth Blackburn, who is a 2009 Nobel Laureate and is one of the scientists who discovered how telomerase actually works. So cells that undergo cell division continue to have their telomeres shortened because most somatic cells do not make telomerase. This essentially means that telomere shortening is associated with aging. With the advent of modern medicine and how far our healthcare system has transformed over the last century, the human lifespan has increased dramatically. And there's an increasing demand for people to look younger and have a better quality of life as they grow older. And in 2010, some scientists actually found that telomerase can reverse some age-related conditions in mice. This may have potential in regenerative medicine. Telomerase deficient mice were used in these studies, And these mice have tissue atrophy, stem cell depletion, organ system failure, as well as an impaired tissue injury response. Telomerase reactivation in these mice caused extension of telomeres, reduced DNA damage, reversed neurodegeneration, and improved the function of the testes, spleen, and intestines. So telomere reactivation may have the potential for treating age-related diseases in humans. Let's say we don't have the machinery in place to detect replication errors, and that sometimes happens. Sometimes uncorrected replication errors do occur, and those result in mutations. So mutations are changes in the nucleotide sequence of DNA strands, and mutations can lead to changes in the protein sequence that's actually encoded in DNA. So on the next couple slides, we're going to talk about the different types of mutations, whether mutations are point mutations or frameshift mutations. So there are several types of mutations. So the first one we're going to talk about are point mutations. The first type of point mutation is known as the silent mutation. So a silent mutation has no effect on the protein sequence. And you see a silent mutation in this position right here. So this T is changed from this original A. So GTA and GTT still code for valine, so there's no effect. We still have serine, valine, proline, tyrosine, serine, valine, proline, tyrosine here. So no change in the protein sequence. The second type of point mutation is known as a missense mutation. So in a missense mutation, this actually results in an amino acid substitution. So we see this A here is changed from this original C here, and there is an amino acid substitution. So this threonine group here is now created where it was originally a proline group. So originally the sequence was serine, valine, proline, tyrosine, and now it became serine, valine, threonine, tyrosine. So a missense mutation results in an amino acid substitution. And the third type of point mutation is known as a nonsense mutation. So nonsense mutations, what they do is they substitute a stop codon for an amino acid. So we see this G right here replace this C here. And several stop codons I need you to be aware of. So I always remember it using this little mnemonic. So UAA, UGA, and UAG. So again, I substituted uracil in the case of the RNA sequence, but in the case of the DNA sequence, all of these U's are going to be T's, okay? So that's what we see here. So we see a TAG, and that's the one down here. Uracil is replacing it in the RNA sequence. So you are away, you go away, you are gone. That's how I remember all the different types of stop codons. So in nonsense mutations, we remove the original coding amino acid and we replace it with a stop codon. So we have serine, valine, proline, tyrosine, and then it changes to serine, valine, proline, and a stop codon. So the DNA sequence is actually causes this stop codon here, which stops and truncates the sequence right there. It no longer reads. And then we get frameshift mutations. So there are insertions and there are deletions with the frameshift mutations. So this may result in the shift in the reading frame or insertion of a stop codon. And that's exactly what we see here. So we see serine, valine, proline, tyrosine, and we have a shift in the sequence. So it goes serine, valine, leucine, leucine. And chromosome mutations can also include translocations, inversions, fusions, and duplications. This is a really good TED Talk, so at this time, please pause the PowerPoint and make sure you listen to this and check this out. Let me know what you think here about the Neanderthal genome research. So this slide should be a review slide for you all, and we're just going to mention some key characteristic differences between eukaryotes and prokaryotes. So again, a eukaryote is going to have a well-defined nucleus where the DNA information is going to be found. In contrast, in prokaryotes, the chromosome lies in the cytoplasm and the DNA is found in the cytoplasm in a particular structure known as the nucleoid. So how do we compact all of this genetic information into our body? If we have these long double helixes just running rampant into our body, wouldn't they just be spurting out everywhere? Well, what actually happens is there is a massive amount of compaction of the eukaryotic chromosomes that allows for all of this information to be retained in our cells. So the DNA double helix is wrapped around an octet of histone proteins to form what is known as nucleosomes. The nucleosomes are then coiled up to form a solenoid shape. The solenoid is further looped back and forth and attached to scaffolding proteins. And then the fully compacted DNA and packaging proteins form a condensed eukaryotic chromosome. So how do we tell what types of animals have what types of DNA in them? Well, we run what is known as a gel electrophoresis at the end of our polymerase chain reaction. I used to do this technique a lot when I was in graduate school. So we start off with DNA extraction and purification. So what we actually did is we took a small snippet of the mouse's tail and we would extract DNA and purify the DNA from the tail. After that, we would run it through the polymerase chain reaction, or PCR. And what PCR does is it basically creates an amplified segment of DNA in order to visualize later on via the process of gel electrophoresis. And that's what we see here on this slide. So DNA has a negative charge and is always going to move towards a positive pole in an electrical field. So this is the negative end and this is going to be the positive end down here. And so what happens is the DNA fragments are going to be loaded in these wells at the top of this polyacrylamide gel. And then it's going to be electrified. It's going to be in a tank with a buffer, and then electricity is going to cause the DNA fragments to migrate down towards the positive end. So in the first lane here, we see a DNA ladder. And the ladder is just used for visualization purposes. And I would assume this is about a 500 base pair ladder. Or sorry, a 100 base pair ladder, because each of these sequences would give us 100 base pair differences. So this would be the 100 base pair marker, this would be the 200, the 300, the 400, and then the 500 is usually more of a hyper color. So this is the bright white one. So this is going to be the 500 base pair marker. And what we see is our DNA fragments here, they range from about the 500 base pair marker all the way down to the 300 base pair marker. And the larger fragments are going to be at top of the gel. So the 500 base pair here is going to be a larger fragment than the 300 base pair one that we see down here. So this gel acts as a sieve sorting fragments by size. So we see nice separation here. So we can clearly see that this is a 400 base pair fragment and this is a 300 base pair fragment. So again, the smaller fragments are going to move more easily through the pores of the gel than the larger ones are. So that's why we have the 500s on top and we have the smaller 300s on the bottom. So the result is that the smaller fragments move faster than the larger fragments sorting by size.